Hello everyone, welcome back to the class. In our previous class, we completed cell cycle part 1 with replicative senescence as a small topic at the last, and we started the discussion of mechanics of cell division. That in eukaryotes there were two cell division which we discussed yesterday, that is mitosis and meiosis. In mitosis, the parent cell divides into two daughter cells, and each daughter cell receives an exact copy of the chromosome in the parent cell, and it is therefore known as equational division. And in other partitioning process, that is meiosis, the genetic material is precisely halved so that fertilization will restore the diploid complement. So that was meiosis. And also we started off the stages of M phase pertaining to mitosis division. Mitosis is the process that partitions the newly replicated chromosome equally into two daughter cells. The term mitosis was derived from the Greek word meaning thread. It was introduced by Walter Fleming. It involves one round of DNA replication followed by single round of chromosome segregation and generates two genetically identical daughter cells. The stages of M phase is prophase, metaphase, anaphase, telophase, and it ends by cytokinesis. Prophase, in that pro means before, meta means mid, ana means back, telo means end. And we were about to discuss about prophase and we ended till that. So today we'll be discussing about prophase, the substage of M phase, that is prophase. So prophase, in this prophase is known to be the longest phase of mitosis. Prophase. So this is the first substage in M phase, that is mitosis. Here it is a longest phase of mitosis, and here in prophase the chromatin which was diffused in interphase gets slowly condensed into well-defined chromosome. Each chromosome has duplicated during preceding S phase. It is duplicated during the preceding S phase and consists of two sister chromatids. Each of these sister chromatids is held together at a constricted region that is known as centromere. It is a constricted region, that is centromere. Each chromatid contains one of the two new daughter DNA molecules. At centromere, the sister chromatids are kept. So this attachment between the centromere and the sister chromatid arms is done, or it is responsible by a member of SMC protein. SMC, what is the full form? SMC, we already discussed this, that is structural maintenance of chromosome. So particularly there is a member of SMC protein which are responsible to hold together the sister chromatids with the centromere, that is cohesin. So cohesin is a SMC protein. Okay. This cohesin is a protein complex. It is a protein complex which is made up of 4 subunits. How many subunits? 4 subunits. Among these 4 subunits, Two subunits are SMC1 and SMC3. These two have ATPase domain. These two have ATPase domains. Okay, this is SMC1 and this is SMC3, and there is two additional subunits, that is SCC1 and SCC3, which connects the ATPase domain. This connects the ATPase domain and forms the ring structure. So which are the subunits present in cohesin? That is SMC1, SMC3, which has ATP domains, and the other two, that is SCC1 and SCC3, which connect the ATPase domains and forms a ring structure. Prophase ends with prophase. First it starts with what? Chromatin condensation. Chromatin condensation into fine, well-defined structure. This is, it starts and ends at disappearance of nucleolus. This is at the end. So prophase ends with the disappearance of nucleolus, and also there is another procedure taking place, that is disruption of nuclear envelope. So it starts by chromatin condensation and ends by disappearance of nucleolus and disruption of nuclear envelope. So this destruction of nuclear envelope is a complex multi-step process. Okay, the nuclear envelope will break down into membrane vesicles. These membrane vesicles are indistinguishable from bits of ER, that is endoplasmic reticulum. So this breakdown of nuclear envelope is a complex multi-step process that begins when, mCDK. What is the purpose of CDKs? So it phosphorylates several subunits. In this case, this phosphorylates NPC, nuclear pore complex, present in the nuclear envelope. So this phosphorylation initiates disassembly of nuclear pore complex. mCDK also phosphorylates the components of nuclear lamina. It also phosphorylates nuclear lamina, which is the structural framework that lies beneath the envelope. So this phosphorylation of lamina components and other proteins leads to disassembly of the nuclear lamina. So here there is disassembly of NPC by phosphorylating NPC, that is nuclear pore complexes, by mCDK, and disassembly of NL, that is nuclear lamina, by phosphorylating nuclear lamina, components of nuclear lamina. Okay, the phosphorylation of both these will lead to complete breakdown of envelope membrane into small vesicles. So in most cells, disruption of the nuclear envelope marks the end of the prophase of mitosis. Okay, however, this disruption of nuclear envelope is not necessary. It is not an universal feature of mitosis, and it does not occur in all cells. So there are now types of mitosis. There are types of mitosis, that is open mitosis and closed mitosis. Closed mitosis. So in some lower eukaryotes, the nuclear envelope remain intact and chromosomes migrate to opposite poles. So nuclear envelope remain intact and chromosomes migrate to opposite poles, opposite poles within the nucleus. So this is termed as closed mitosis. It is occurring in lower eukaryotes like yeast, diatoms, etc. In cells of higher eukaryotes, it undergoes open mitosis, that the nuclear envelope breaks down. Nuclear envelope breaks down at the end of prophase. So this is happening in higher eukaryotes. So this is the major difference between open mitosis and closed mitosis. Towards the end of prophase, we know there is disappearance of nucleolus and breakdown of nuclear envelope, and there is another one major important factor at the end. It is seen at the end of the prophase, that is the cytoplasmic microtubule. Cytoplasmic empty microtubule that are a part of interphase cytoskeleton. This cytoplasmic microtubule disassemble and there is a formation of mitotic spindle formation at the end of prophase. So this is mitotic spindle plays a vital role in metaphase stage. So therefore it is priorly formed in the prophase stage. So now what is this mitotic spindle? Mitotic spindle. So mitotic spindle is a bipolar structure. Having both plus and minus ends. So it is composed of microtubules. Plus associated proteins, and this mitotic spindle initially assembles. So it initially assembles outside the nucleus. Between separating the centrosome. So mitotic spindle is a bipolar structure. It is made up of cytoplasmic microtubules and and associated proteins. It assembles outside the nucleus initially. And it, as I said, it is a bipolar structure. It is having both plus end and minus end. So mitotic spindle helps in nuclear division. So it mediates nuclear division, which separates the chromosome also. Spindles are microtubules, each of which is a polarized tubulin polymer. It is a polarized tubulin polymer. Polarized means having particular pole, charged pole. Okay, one end of this tubulin polymer is minus end and another end is plus end. This minus end, so-called minus end, is embedded in spindle pole. And the other end, that is plus end, it is pointing outward from pole. So consider this as a pole. Okay, this as a pole. So minus end are projecting towards this and plus end are outward from the pole. Okay, the mitotic spindle is of 3 types. So mitotic spindle are of 3 types. That is, first one is kinetochore microtubule, polar microtubule and astral microtubule. So these are the 3 types of mitotic spindle: kinetochore microtubule, polar microtubule, astral microtubule. I'll draw a diagram now. See, there is a cell. Here. Consider it as an animal cell. And we have poles. Here. 2 poles we have, that is MTOC. We'll name that as MTOC. Okay. I'll write it again. So we have a cell, animal cell. Consider it as an animal cell, and we have 2 poles. That pole is a centrosome, which is MTOC with centrioles. So these poles are MTOC, that is centrosome, MTOC with centrioles, from where the microtubules is what we can say, it is spread, it is exerted out, it is pointing outward. Okay, so there are thread or the lines like this, you can see. Okay, which is towards outside. So these are plus ended or plus ends are towards what? Towards outside, towards the external side. So it is opposite to the pole. So it is not towards the pole, it is away from the pole. So there is another line we can draw. So first I'll draw chromosome. I'll draw a chromosomes here. Okay, to which there is a kinetochore. To which the spindle microtubule is attached. Okay, and the one which is exerting out is a minus end. So there are other lines or other microtubules exerting outward. There are other microtubules exerting outward, and that exerting outward, that point is plus end. That point is plus end. In this we can mark all three types of microtubules, that is kinetochore microtubule, polar microtubule, astral microtubule. First we shall label the kinetochore microtubule. Microtubule which is attached to a specialized protein complex, that is called kinetochore, is known as kinetochore microtubule. See, this line is attached to this microtubule. So this microtubule is KMT, kinetochore microtubule. So the end, the plus end, is attached to the specialized protein complex called kinetochore, mature on the centromere of each chromosome. The plus end of kinetochore microtubule, so the plus end of microtubule remains attached with the kinetochore. Okay, next, polar and its function is, at anaphase it pulls the two sister chromatids towards opposite poles. So it pulls out the sister chromatids towards opposite poles. Okay, next one is polar microtubule. We call this as interpolar microtubule also. Okay, this will not interact with any chromosomes but overlap with polar microtubules from the opposite poles. So here, which microtubule is not attached to the chromosome? This and this, this and this. So but even if at all it has to interact, it interacts or it overlaps with the polar microtubule from the opposite side. So it is interacting with this, this type of microtubule is known as PMT, polar microtubule. It is also known as interpolar microtubule. So there is another at the center, I'll draw, then you will get to know. So this is plus end and this is plus end. This is also a polar microtubule which is not interacting with chromosomes but overlapping with polar microtubule from the opposite poles. And at the last type, that is astral microtubule, which form the aster. So it radiates outward from the centrosome. So centrosome is made up of centrosome matrix and with pair of centrioles, so which contributes to the formation of microtubules and mitotic spindle. So it extend or it radiates outward from the centrosome towards the cortex. So this, as I mentioned here, plus which is radiating outward from the pole, from the centrosome, that radiating it towards the cortex. So this type of MTs is known as AMT, astral microtubule. So the centrioles which are associated with the centrosome matrix will form a centrosome, okay, and are not required. So these are not required for the nucleation of spindle fibers. So in plant cells, so there is absence of this centrioles altogether, but the nucleation of spindle fibers occurs. So it is therefore thought that centrosome matrix, which contains a set of protein, is very important for nucleation of the spindle fibers. So this is animal cell where you can see what the aster, or you can say centrioles. So in animal cell, the aster is present, astral microtubule is present, and the centrioles is absent. So this is which cell? This is animal cell. In animal cell you come across aster, you come across centrioles at the pole, centrosome. So that centrosome is acting as a pole. It is made up of centrosome matrix plus centrioles, Cl, centrosome matrix and centrioles. Okay, now I'll draw another cell. Okay, another cell. This is another cell. Particularly it is a plant cell. In plant cell, how do these microtubules are present? So as we know, there is no centrioles. Okay, there is no centrioles, but there are other two microtubules, that is polar microtubules and kinetochore microtubules. I'll write the chromosome to which kinetochore is present. Okay, kinetochore is present at the centromere, and the microtubule which forms connection with these kinetochore is known as KMT, kinetochore microtubule, and the other one which do not interact with chromosomes but overlap with polar microtubule is polar microtubule, or it is known as interpolar microtubule. The charges are same. The charges are same as you saw in animal cell. So this part is known as MTOC, where there is lack of centriole. So this type of cell is known as, or this features is present in plant cell. So let us see one by one now. Prophase is where chromatin slowly condense into well-defined chromosome. Each chromosome has duplicated during the preceding S phase, and now it consists of two sister chromatids. Each sister chromatids held together at a constricted region, that is centromere. Each chromatin contains one of the two new daughter DNA molecules at centromere. Sister chromatids kept together by the member of SMC family of protein, that is cohesin. Particularly, cohesin is made up of 4 subunits. 2 are SMC1 and SMC3 with A2 ATPase domain, and 2 additional subunits, that is SCC1 and SCC3, connects to the ATPase domain, forming a ring structure. So prophase ends with the disappearance of nucleolus and disruption of the nuclear envelope. So nuclear envelope breaks down into what? Into membrane vesicles that are indistinguishable from bits of ER. So cytoplasmic microtubules that are a part of interphase cytoskeleton, they disassemble and mitotic spindle begins to form. This mitotic spindle is a what? Bipolar structure composed of microtubules and associated proteins. So the spindle initially assembles outside the nucleus between the separating the centrosome. So this is about the nuclear envelope breakdown. So it is a complex multi-step process. It begins with mCDK phosphorylating the several subunits of nuclear pore complex. So here there is phosphorylation of nuclear pore complex protein and also phosphorylation of lamin proteins, nuclear lamina, components of nuclear lamina, which is helping for the disassembly of the nuclear lamina in the prophase stage. Okay, it at the early telophase there is dephosphorylation of lamin protein. Before that the phosphorylation of phosphorylation of lamin and NPC protein, see the arrangement, that is an interphase. At the end of the prophase it is phosphorylating and it is responsible for disassembly of what or nuclear envelope break down. So in cells in cells of lower eukaryotes like yeast and diatoms, the nuclear envelope remain intact and chromosomes migrate to opposite poles within the nucleus. That is termed as closed mitosis. So this is open mitosis where the nuclear envelope breaks down. And next we discussed about the mitotic spindle. The nuclear division is mediated by a microtubule based mitotic spindle, and this mitotic spindle is formed in the prophase stage, and it has a major function to play in metaphase stage. It separates the chromosome in metaphase. Spindles are made up of microtubules, each of which is a polarized tubulin polymer. Polarized in the sense they have a particular charged pole with one end, so it is so called minus end, which is embedded in the spindle pole, and the other end plus end is pointing outward from the pole. Here you can see all plus are outward and minus are towards the pole. There are three types of mitotic spindle: kinetochore microtubule, which is which is the microtubule which is attaching the kinetochore; polar microtubule, which do not interact with any chromosomes but they overlap with the opposite polar microtubule; astral microtubule are exerting or radiating outwards towards the cortex of the cell. Kinetochore microtubule attaches with specialized protein complex called kinetochore. A plus end of kinetochore microtubules remain attached with the kinetochore. At anaphase stage, it pulls the two sister chromatids towards opposite poles. Polar, that is, that do not interact with chromosome but overlap with the polar microtubule from the opposite poles. Astral pole forms a aster which radiate outwards from the centrosome, which is made up of centrosome matrix associated with a pair of centrioles, towards the cortex. So plant cells lack centrioles altogether, but the nucleation of spindle fibers occurs. So therefore thought that the centrosome matrix, which contains a set of proteins, is very important for nucleation of spindle fiber. So now there's the information about kinetochore here. Kinetochore assembles at the centromeric region. Here you can see kinetochore, which is present at the centromeric region. It is a giant multi-layered protein structure. It has three layers: inner kinetochore, which forms the interface with chromatin; outer kinetochore, which forms the interaction surface for spindle microtubules; and central kinetochore, which is the region between the inner and the outer kinetochore. What is centromere now? It provides the foundation for assembly of the kinetochore. So the centromere of the human chromosome are heterochromatic region, and that consists of repeated sequence known as alpha satellite DNA. This chromosomes lacking a localized centromere and primary constriction sites are also called as holocentric chromosomes. In these chromosomes, kinetochores diffuse and kinetochore microtubules attach along the length of the chromosome. Here there is attachment of the mitotic spindle, attachment of chromosome to the mitotic spindle. In late prophase, see, there is no attachment. Okay, there is what in late prophase, the mitotic spindle poles have moved to opposite sides of the nuclear envelope, but there is no attachment to the kinetochore. Okay, so in early prometaphase, lateral kinetochore attachments are seen, chromosomes arms pushed outward, and in mid prometaphase, end on attachment is found. In metaphase, by orientation. So this is already discussed. So how the kinetochore attachment, alternative forms of kinetochore attachment, which is unstable, which is stable, which has already discussed under under the error correction mechanism mediated by kinetochore, that is error attachment, that is amphitelic, which is correct attachment, monotelic and syntelic. Okay, this is unstable, this is unstable, this is stable, that is amphitelic, where sister kinetochores are anchored to the microtubules from the opposite poles. So that's it for today. In our next class, we'll be discussing about metaphase, the second substage of M phase. Thank you.